obviously, if you can't sense what's going on inside or outside of the cell, cell expression and gene expression regulation really doesn't make a whole lot of sense. So intracellular and extracellular sensing mechanisms are really essential to cells to be able to know what genes to express. So you can't respond to the environment if you can't detect what's actually going on inside and outside. And so inside the cell, it's relatively straightforward, at least if we don't look at it too far in depth. But you'll have these regulatory proteins that can bind ligands, which are typically associated with metabolites, that are specific to each regulatory protein to either activate or repress the genes. So this can happen based on what is actually happening in the cell, and we're going to look at this a lot more in depth in upcoming videos. Getting signals from outside of the cell is a little bit more complicated because you basically have to have some sort of ability to sense what's happening on the outside and relay that message to what's happening on the inside. So usually this is associated with these kind of transmembrane proteins that act to send those signals in. And the most common example for this is called the two-component signal transduction system. And what this does is basically you'll have what's called a sensor kinase in the plasma membrane. And so remember, a kinase is an enzyme that transfers a phosphate group from ATP or a phosphate group from some compound to another compound. So kinases are associated with phosphorylation. And so with the sensor kinase, what's going to happen is you'll have this sensory domain on the outside and this kinase ability on the inside. And so based on this signal, you'll have the ability to autophosphorylate, meaning you'll take an ATP molecule and kind of strip the phosphate from that on this kinase domain of the sensor kinase protein. This can either then directly transfer that phosphate group to the response regulator, or you can have what's called a phosphorelay, where a number of other compounds get phosphorylated that will ultimately trigger the response. So now you have this response molecule, this response regulator, that can go on to activate, repress transcription, and interact with things in the cell. And so one thing that's interesting to note, we're going to talk a lot about ligand binding to specific regulatory proteins, but this is different. This is actually a covalent modification. So anytime you're adding a phosphate group, that is covalently modified and a little bit more permanent than just something loosely interacting like what we'll see with the ligands. And so this has to be turned off by a phosphatase. Sometimes this can actually be done on the same sensor kinase that provided the signal transduction from the outside of the cell anyway. So you basically have to have another enzyme that will actually cleave off that phosphate group to kind of stop that signal from happening. So this is relatively more permanent changes in the cell.